We'll have a cancer and chronic disease session now, and we have three speakers, Dr. Dasgupta, Professor Partha Majumder, and Dr. Murim Choi. So they're going to speak on early liver disease, and then oral cancer, and then in NAFLD, non-alcoholic fatty liver disease. So let me introduce Dr. Dasgupta. He has a stellar career. He had a postdoc from Harvard Medical School and then joined New York University. Spent a lot of time before moving to GIS. He leads a program in precision oncology and cancer evolution. He had worked in different domains of health and disease, both in the healthy atlases and also in the cancer atlases. So, namaste.
Thank you so much, Don, for a very generous introduction. And good afternoon, everybody. Hope you had a good lunch. And I'll try to keep you from falling asleep, hopefully. All right. Let's see. All right. And yeah, a huge thanks to the organizers for the opportunity to speak. Mainly I'll talk about stuff that we are currently doing, not so much a detailed dive into the data per se, but the kind of data that we are trying to generate and how I think as a community at HCA Asia we could really combine our efforts to look at some of the questions that I'm going to talk about. So fundamentally we are interested in cancer evolution and more recently really getting interested in understanding how tumors evolved in the context of their microenvironment, and which we hope will uncover the true potential of using combinatorial therapeutics, combining, say, immune checkpoint inhibitors with other targeted inhibitors that specifically target the tumor microenvironment. In terms of the approach, well, no surprise here. We are at a single-cell meeting. We use a variety of single-cell omics, so single-cell RNA-seq, ATAC-seq, combined with the very original single-cell approach of multiparametric flow cytometry to really map the trajectories of tumor evolution both in time and space. So we try to use longitudinal samples or cross-sectional samples whenever available, and then do validation using spatial. And while the hypothesis generation is mainly with patient samples, we try to get into some of the functional interrogations using mouse models, which then can be clinically validated again using different techniques. We work on a variety of different solid cancers, mainly focused on GI tract as well as hepatobiliary. For today's talk, I'll mainly focus on some of the work on liver cancer. It is a major burden, especially in Asia, and there is an alarming rise in incidence worldwide, with the new HCC cases projected to be increased to increase by about 55% by 2040. The other thing to remember is HCC is often a result of a distinct etiologies of progressive liver disease, and the two major causes is either viral hepatitis, so patients with chronic hepatitis B, and more recently fatty liver disease, which is NAFLD and NASH. Although based on the current EASL guidelines, we are all supposed to be calling it MASLD, which is a metabolic dysfunction associated steatotic liver disease. It's a mouthful, but it's something that I think at HCA Asia we should start thinking about. Previous work from the lab aimed at generating a single-cell atlas of HCC revealed this remarkable fetal-like reprogramming of the tumor microenvironments that remodels the vasculature and the immune cells to establish immune tolerance. This is in fact work done by Ankur when he was a postdoc in my lab, and has since started his own lab in Australia. And more recently, in collaboration with Valerie Chu's lab, we also showed that canonical Wnt pathway, a pathway that has been very close to my heart for a very long time, may be playing a really early role in establishing and promoting immune suppression in the liver tumor ecosystem. An interesting aspect is that when we now start looking at these fetal programs, at the fetal-like programs in early liver disease, what we find is that these programs and immune tolerance may actually represent an earlier event, and it's typically associated with damage-associated activation of regenerative programs. And we find these programs both in cirrhotic liver, so this is just an integration of our own data set with the cirrhotic data set, single cells from Neil Henderson's lab, and more recently, this is a paper under review right now in chronic Hep B, and I'll focus a little bit more a bit later in my talk. We do see these emergence of these fetal or immune suppressive phenotypes in situations of, say, incomplete wound healing or chronic non-healing wound, representing dysfunctional regeneration. However, if you were to have a mouse model, for example, of 2/3 partial hepatectomy, which is representative of complete wound healing, you'll see the increase in some of these fetal-like regenerative programs, but then they subside over time as regeneration is complete. Now, these observations are highly pertinent to the current which I think a major knowledge gap in the clinic, which is to say that the etiology of HCC in fact might be changing. In fact, liver cancer is the fastest rising cancer related to obesity based on metabolic dysfunction and damage to hepatocytes. And specifically for HCA Asia, this is a recent study from 2019. It appears NASH is the fastest growing cause of HCC. The incidence is actually very similar between men and women, so very different from Hep B related HCC, where men get it about 4 times higher proportionality compared to women. And you can see in terms of the rapid increase, specifically in India as well, this is just in the last 9 years. So the proportion of HCCs that are related to NAFLD is on a rapid rise. So therefore, again, becomes really important for us and pertinent for us to study this. Somewhat controversial, NASH HCC is also appeared to be less responsive to immune checkpoint blockade, although the jury's still out there. But fundamentally, the hypothesis that we are working on, and this is again collectively with the liver seed network as well, is that we think that liver damage, so whether it's metabolic dysfunction associated or viral hepatitis, for example, may activate regenerative pathways that can reprogram the liver tissue microenvironment in somewhat of a pro-tumorigenic niche. But obviously there are multiple questions that we want to ask. What are the mechanisms that drive these distinct etiologies of liver disease progression, so whether it's viral or NAFLD NASH to HCC? How do these distinct etiologies relate to altered cellular organization or neighborhoods and their ecosystem? How do they modulate therapeutic response? And better understanding of this, can we then target these mechanisms as early preventative intervention strategies, which has now become the major thrust on my lab. We want to be able to reverse NASH, NAFLD, reverse chronic Hep B, for example, so that we don't get to or minimize the risk to progression to HCC. Once you get HCC, it's already kind of late. And this is a slide that Sham shared earlier yesterday, and this is what we are planning to do. And I'm showing you this again mainly so that it's also an invitation for anybody who's interested in looking at these early trajectories and etiologies, the distinct etiologies of liver disease progression to either NASH or non-NASH HCC. Please come and have a chat with me. But what we are doing currently is basically generating a multimodal, so single-cell RNA-seq, ATAC-seq, as well as flow cytometry-based assessment to generate a spatiotemporal atlas of both liver disease progression as well as regression. We already have some really beautifully curated samples, patient cohorts with distinct stages of NAFLD and early NASH and late NASH, and then their progression to HCC. We also have certain cohorts where we have pre-post samples from patients who were under diet and physical activity regimen, so where they show reversal of NASH, for example. So lifestyle and dietary interventions. So the idea is to generate a comprehensive atlas, not just with single-cell multiomics, but also getting into some of the DESI MSI, which is a metabolic spatial imaging technology. And then again, in parallel, we are also looking at some of the mouse models of liver disease progression for functional studies. However, for the rest of today's talk, so that's all data being generated, and hopefully I can talk to you about it in a lot more detail the next time we meet. But I'll talk to you a little bit about some of our more recent work. This is currently a preprint and under revision of single-cell analysis of chronic Hep B functional cure. Functional cure as it is defined by the loss of S antigen. So why are we interested in Hep B? Well, viral hepatitis and HB can activate just about all 10 hallmarks of cancer, and in fact, one of the predominant cause of HCC, so 40%. Even though NAFLD-NASH is very rapidly on the rise, this is work led by two extremely talented postdocs, Narmada and Ati, together with Niranjan, who's a bioinformatician, and Lim Sengi, who's our clinical collaborators. So why do we care about understanding the mechanisms of functional cure, that is loss of S antigen? And as you can see in this graph, and these are again based on EASL guidelines, there are 4 phases of chronic Hep B, and then functional cure as defined by the loss of S antigen. But the reason we want to understand is that, well, firstly, it's a clinical endpoint that the clinicians want to reach in terms of achieving functional cure, and the reason is that it minimizes your progression to HCC. However, the mechanisms that are driving functional cure in patients who are treated with nucleoside analogs combined with interferon alpha to elicit an immune response are really poorly understood. So we really want to understand those mechanisms that underlie long-term functional cure as a means to then minimize the risk of progression to HCC. And so that's what we did. So this is just the workflow that we have. So it's about 22 patients, 15 with chronic Hep B, 7 that achieve functional cure after treatment with antivirals combined with interferon. We generated single-cell data from core biopsies as well as matched PBMCs. And also analyzed by flow-based immune phenotyping, and then followed by spatial validation. So this is now in Bar Archive. You can read more about it, but hopefully you'll see it soon. So I'll just focus on some of the key observations that we've made in terms of mechanisms or immunopathological cell states associated with functional cure, and what we could learn about how can we affect that functional cure in patients. So first, in functional cure patients, so the patients that have lost S antigens upon treatment, they exhibit a remarkable switch from an immune tolerant to immune suppressive to an immune active environment. So you lose these clusters of Tregs, which are regulatory T cells, as well as lose these clusters of exhausted CD8 positive cells. The other really interesting and very surprising thing is that we also see this gain of this red cluster, and these turn out to be CD4 CTLs. And this is surprising because these are CD4 T cells, helper T cells that acquire properties of innate immune-like cells, including gain some of the markers such as NCR3, but they also gain cytotoxic or cytolytic abilities in terms of expressing granzymes, perforin, and TNF. We could validate this with functional with flow cytometry. Again, you see the CD69, which are the tissue-resident populations of CD8s, now stratified with different levels of S antigen. So red is high levels of S antigen, orange is moderate, green is low, and blue is functional cure. Let me see if this. So you see this loss of the exhausted populations, and moreover, you also see gain in these CD4 T cells that are gaining this marker KLRG, which specifies these terminally differentiated antigen experienced CD4 CTLs. They also express these markers, and we validate that with flow cytometry, as well as CyTOF. And this is again looking at patients with very high levels of S and low levels of S. You can see about a 2.5 to 3-fold increase in granzyme B and perforin in the CD4 KLRG population. So that was the first surprise, right? The second thing that is really interesting, and this is functionality of these CD4 cells. So we basically stimulated these T cells with pooled peptides representing different viral proteins, including X, core, Pol, and S antigen. And what I really want to focus your attention to is that the HBS-specific CD4 T cells, they appear to be present at much higher frequencies than the CD8 S-specific CD8 T cells, right? Suggesting that the dominant S peptide specific response is actually coming from a CD4 effector, not CD8s in functional cure, suggesting that these CD4 CDLs may be actually playing a really important role in not just driving functional cure, but even maintenance of that functionally cured state. And you also see increased multifunctionality in terms of interferon and TNF-alpha expression in these S-specific CD4 T cells, and this kind of corroborates somewhat some of the recently published data from Georg Lauer's group. They used tetramers basically against MHC class II, where they showed that these Hep B-specific CD4 T cells are reliably found in the S-negative patients and almost rarely found in the S-positive. Right. So one thing that we started thinking about, well, then what are these CD4 CTLs recognizing? Because these would be interacting with MHC class II. And the other major surprise that we find in the functionally cured state is this emergence of these MHC class II expressing hepatocytes, suggesting these hepatocytes may be gaining accessory APC, antigen presenting function, upon interferon alpha treatment. Now, a couple decades ago, actually, this has been shown in vitro that interferon can actually induce an MHC class II switch. What we think is that the patients who have undergone this nucleoside analog treatment combined with interferon, they're causing the switch in MHC class II in hepatocytes, which are then recognized by the CD4 CTLs to take them out. However, there lies a conundrum, because we are talking about functional cure, and at least clinically speaking, you were saying, well, I mean, the S loss, there is S loss, right? So at least in the serum. So what exactly are the hepatocytes actually presenting, even if they were to present viral peptides, for example, because this is a S loss case. But what we went back and started looking at specifically in a more granular fashion, the different viral parameters in these functionally cured patients, and what you can see with digital droplet PCRs, as well as single-cell RNA-seq actually. So we took the unmapped reads and mapped it to the Asia-specific genotypes of the virus, and we see a 50 to 100-fold reduction in the production of virus, but the virus is definitely still being produced in the functionally cured state. So you have levels of cccDNA, Hep B DNA, the HBV 3.5 kb mRNA as well. And then if you look at the normalized ratio of the mRNA to the amount of cccDNA, which is transcribing this viral DNA, you see the transcription rate is more or less same, or very similar between chronic Hep B and functional cure. But we could now see the same thing with the single-cell profiling as well. So this is basically mapping the unmapped reads otherwise now to the viral genotype, suggesting that even though there is very low levels of the virus in the functionally cured state, they could still be now presenting the native peptides, as also suggested by Georg Lauer's group, and eliciting a CD4 CTL response. So a couple key take-homes: that the loss of S antigen detection in serum is not necessarily cannot be equated to loss of S transcript production in the hepatocytes, and the reduced viral load upon nucleoside therapy, combined with the interferon-mediated activation of MHC class II and the IL-2 driven differentiation of CD4 CTLs. There's a lot more details there that I'm skipping over because of time constraints, but eventually would result in a better outcome to chronic Hep B. So just a working model for the CHB resolution is that we think that in the state of chronic hepatitis B, you have a defective priming of the adaptive immune system by a lot of pseudoantigens that are floating around, that are produced by the hepatocytes, which then results in chronic antigen stimulation and immune cell exhaustion to create this tolerant microenvironment. And upon treatment with nucleoside analogs and followed with to lower the viral load, and then followed up with interferon therapy, you can reactivate these pro-differentiation of CD4 CTLs together with MHC class II, where hepatocytes are now presenting native peptides to CD4 CTLs. And again, a really important role that didn't really talk about here, of the innate immune arm as well, with NK cells and neutrophils, we think are really important in affecting resolution of Hep B. But overall, the other point just wanted to make is that probably need to consider CD4 T cells when designing the next generation of immunotherapies. So just a couple last take-home messages or take-home slides that I will leave you with. So at this point, what we are now trying to do is kind of develop a blueprint for across time span and disease, both in health as well as disease states in the human liver. So this is basically a Skivi-based integration of the different data sets that we have now downloaded or generated, and now we are combining them to generate these integrated maps, and we can annotate these maps. But fundamentally, we want to kind of try to see, like, these are the disease-associated cell states, for example, in the fibroblasts, which are not there. Perhaps, okay, this is Mazhanifa's fetal data sets, so early versus late. We see very interesting changes in endothelial cells in early versus late in the fetal liver, and some of them could be represented again in situations of chronic liver damage, whether it's cirrhosis, Hep B, or HCC. Similarly, we are also developing a blueprint for healthy and disease states in using the mouse models. And again, hopefully you could see this with control diet. For example, this olive green cluster is basically a fibroblast cluster, and you see remarkable changes, what's happening in the fibroblasts, as well as the endothelial cells and the hepatocytes, which are very distinct from what we see in the animals on normal chow or normal diet, suggesting that there is a remarkable remodeling of these different parenchymal and the non-parenchymal cell states, and it's really the crosstalk between these that then generate, what we think would be a pro-tumorigenic microenvironment. So with that, I'll end here and to the most important slides, really, of acknowledgement. Patients who were so generous with donating all their samples for research. Really, the true heroes are the people who do the work. I mean, these are the DG Lab heroes, the current and the past members. I'm basically the cheerleader on the sidelines, and amazing collaborators and clinicians out there. And while we are doing all this work, we also have a lot of fun. So happy to take questions. I'll end here. Thank you.
Hi Ram, great talk as usual. So I was curious, seeing this story evolve in your lab, have you also, what are your observations regarding interaction of these particular oncofetal reprogramming cells with the malignant cells in the tumor ecosystem?
That's a great question. So we do, this is something that we are looking at very carefully. So we do have certain hepatocytes, right, that may be interacting with specific populations of endothelial cells. We think that there are certain endothelial cells, or the nature of these endothelial cells also help them interact with certain kinds of fibroblasts, right, and these could be involved in either pro-remodeling or immune modeling fibroblasts, which then eventually affect recruitment of different immune populations. And then you have different chemokines, cytokines that are produced, which can at least recruit certain kinds of immune cells or white blood cells, and then perhaps, reprogramming them into different either tolerant or immune active states. But it's not, so the question is, of course, that the reason we are doing this temporal cross-sectional analysis, we do not really know what comes first, right? Usually we've been just looking at snapshots. So hopefully once we finish this, once we complete this analysis, we'll have a much better idea about the temporal sequence of events of these interactions to figure out what, we could potentially target mechanistically as well. Yeah.
There's a lot of questions about, we might not have time for everyone, so maybe one more from the other microphone, and then we can take the rest up at the break.
Okay, a fantastic talk. So my question is about chronic functional cure. Functional cure of the chronic hepatitis B. So you said CD4 T cells become activated, they become CD4 CTLs, and also in the scheme, so you show that activate CD4 T cells provide help to the B cell to make antibodies. So do you see the upregulation like T follicular helper-like genes upregulations like L21, 613, something like this in a functional cure?
Really good question. Actually, we do see some evidence of the follicular helper cells that appear to be highly enriched in functional cure. We're trying to understand that better. There is also a very clear clonality enrichment of the B cells as well. And so we are trying to clone them now and look to see if we could identify specific BCRs basically that may be involved that we could potentially then be used as immunotherapies. Yeah.
Thank you.
Thank you.
Okay, let's thank Ram.
Thank you very much.
So it is a great privilege for me to introduce our next speaker, Professor Partha Majumder. He is a National Science Chair of Government of India. He is a distinguished professor of J.C. Martin Center for Liver Disease, and he is the founder director of India's only biomedical genomics institute called National Institute of Biomedical Genomics. So by training, he is a statistician at core, and he is elected member of all national academies of India, TUAS, and many of these local academies. So, Professor Majumder, the floor is yours, and he's going to talk about oral cancer atlases.
Very generous, Nidhan. Thank you very much. I'm going to talk about oral cancer of a particular kind, and I'll tell you why I'm restricting myself to a particular kind of oral cancer or cancer within the oral cavity. This is the reason why we look at oral cancer is primarily because the prevalence of oral cancer and the incidence of oral cancer is very high in India. It's a #1 killer among males in India, and that's the reason why we focus on this. The site distribution of oral cancer is very interesting. It's exactly the opposite in India from what is found in the West. In the West, it's predominantly tongue cancer. About a quarter will be cancers of the other parts of the oral cavity. In India, it's essentially other parts of the oral cavity, only about a quarter will be tongue cancer. The other parts of the of the oral cavity is called gingivo-buccal oral cancer. So I'm going to focus on gingivo-buccal oral cancer, and I'll contrast it with tongue cancer at some stage. The hazardous combination is just this. We know about the chewing of tobacco, etc., and that's the reason why the prevalence is very high, because that's very causative. I'm going to run out of time for sure, and so let me acknowledge the funding agency. All of this work was done with funding from the Department of Biotechnology, Government of India. I have a bunch of people, a bunch of clinicians who actually collaborated with us over a long period of time, from ACTREC, from Tata Memorial Hospital in Mumbai, Tata Medical Center here in Kolkata, and more recently for the single cell work with our Ahmed Dental College and Hospital. My collaborators in terms of the basic science or genomic sciences have been Arindam and Nidhan. And the people who actually did the single cell work are all, most of them are here and have given talks. Silarine, Shumitabo, Arunima, and Shomnath. Shomnath isn't here, but he was one of the early initiators. I'm going to summarize in very briefly what we found out about oral cancer over a period of time using bulk sequencing. So it's going to take 2 or 3 minutes. So let me share my slides. So essentially 10 genes drive oral cancer, and this is based on a sample of about 500 patients. And these 10 genes, as you can see, they're ordered in order of their involvement in oral cancer, P53, caspase 8, FAT1, and so on and so forth. Most of these act as tumor suppressors. Some of them can act as oncogenes or tumor suppressors. In the context of oral cancer, they all, most of them act as tumor suppressors, which is really not good news. When we compare our data with the data from the West, which is predominantly tongue cancer, what one finds is that there are certain genes that are common, but there are also certain genes that are specific to gingivo-buccal oral cancer, and I'll focus on some of these genes at a later point of time. Also, even among the common genes, caspase 8 frequency, as you can see, is extremely high in gingivo-buccal oral cancer compared to tongue cancer. We published this some years ago. We looked at metastasis, and there are oral cancer patients who will not develop metastasis for a long period of time, and there's a subset of patients who develop metastasis very rapidly. And what we essentially found out is that it's predominantly because of genes in the homologous recombination pathway that are altered as a result, and there's also a huge amount of chromosomal instability in patients who come down with lymph node metastasis in a short period of time. Again, we published this. We looked at the evolution of oral cancer from a precancerous stage. The precancerous stage is called leukoplakia. There's also erythroplakia and leukoplakia. We looked at essentially leukoplakia, and from each patient we sampled blood, tumor, leukoplakia tissue, and adjacent normal tissue. So we did a comparison of all of this, and essentially what we found was the progression from a precancerous stage to frank cancer was the caspase 8 genes were initially altered, followed by Notch1 and P53, followed by the other kinds of problems such as EGFR amplification, chromosomal instability, and so on and so forth. So this understanding from pre-cancer to cancer actually was very instructive for us, and that was one reason why we could explain why there is such a huge frequency of caspase 8 mutations or caspase 8 gene alterations. We also looked at recurrence and prognosis. So in a subset of patients, the cancer will recur in a short period of time, and the prognosis will be bad. Again, we essentially found two genes. Those are the two genes, including CD73, that's overexpressed in the tumor tissue compared to normal tissue. And these overexpression of these genes in the tumors essentially leads to a bad prognosis. We looked at immune repertoire reconstructed from bulk RNA sequence data. Most of them were BCRs, and about 10% were TCRs. And the bad prognosis was essentially associated with reduction of clonotype diversity, which again supports higher immune infiltration and better antigen recognition, resulting in better prognosis. Published this. So I'll come to, so essentially the take-home message from all of this is that there is a huge amount of heterogeneity, genetic heterogeneity or genomic heterogeneity, both in terms of just cancer as an entity, or if you look at other phenotypes such as progression and so on. So we wanted to assess the level of heterogeneity at the single cell level, and so we embarked on this, and essentially we had a set of 12 patients. These patients actually express genes, the number of genes expressed is by and large the same. The expression levels are highly correlated between bulk and a single cell. We sampled about 29,000 cells, and these 29,000 cells formed 16 clusters. Each cluster expectedly comprised cells from multiple, essentially mostly all of the patients, but the proportion of these cells in these various clusters from each patient was highly diverse. Expression of cell type, representative genes. So these are the cell types that represent these clusters, and those are the cluster identities. There are certain genes that are expressed in multiple clusters, but in varying proportions, as one would expect, and there are certain clusters that essentially express one single gene. And based on these marker genes, we of course identify what kind of cells are there in that particular cluster. We wanted to identify the malignant cells. Malignant cells are essentially, these are epithelial cells. Malignant cells are essentially identified by copy number variations, and so infer-CNV is generally used to identify malignant cells. We were not completely happy with infer-CNV for reasons that I do not have the time to elaborate on. So essentially what we did was to form a reference level of genes for the non-epithelial cells, and then using that as a reference for each of the epithelial cells, then we went ahead and found a distance between that reference sequence, reference expression, and the level of expression of these genes in the epithelial cells. When we plotted the distance, that was just the Euclidean distance that we calculated. When we plotted the distance, we found a very nice bimodal distribution for every patient. Essentially, the antimode was mark the difference between the malignant and the non-malignant cells. We, of course, the borderline cells we excluded, and these were the malignant cells, and those were the non-malignant cells. Again, we looked at the expression levels of the particular genes to identify which of these, peaks or which of these areas comprise the malignant cells. So we called this the distance profiling method. Again, we compared it with the INFER-CNV, and we found that the distance profiling method works actually better. Anyway, we use both INFER-CNV and our method in order to identify the malignant cells. The non-malignant cells, essentially, all of these cell types were present in all individuals. There was significantly higher infiltration of immune cells, B, T, myeloid cells, and so on. The major cell types in the tumor microenvironment are T and myeloid cells. This is what we identified. We found in these 12 patients there were about 7,500 malignant cells, and those are the clusters. We identified 16 clusters in these among these patients, or in the class of 7,500 malignant cells. Interestingly enough, the clusters form two groups. As you can see, two groups of clusters, and we were intrigued by these two groups of clusters, and then when we went back and looked at these two groups of clusters, we identified that this first group of clusters came from a patient, all of whom had oral submucous fibrosis, and the other patients did not. So it was really the phenotype, the oral submucous fibrosis, which is essentially a precancerous lesion, but very close to cancer. Many of them actually develop cancer. So these are all cancer patients who also had oral submucous fibrosis, and that's the group 1 patients, and the group 2 patients are oral cancer patients without submucous fibrosis. Most clusters were interestingly patient specific. Only 5 of these 17 clusters were from multiple patients. These, as I said, that there were 3 oral submucous fibrosis patients. The group 1 clusters were all from these 3 patients. The remaining clusters, the group 2 cluster, were predominantly from the remaining 9 patients. There were 3 genes that actually discriminated between the group 1 clusters and the group 2 clusters. Those genes are FOS, ATP1A1, and DSP1. And we did a discriminant analysis using these 3 genes, and what we found was that these 3 genes, levels of expression of these 3 genes completely discriminated the 2 clusters, distinguished the 2 clusters. That was kind of interesting. Most of the clusters, as I said, were patient-specific. The specific number of cells, about 41% of the cells we identified undergo EMT or partial EMT that results in tumor invasion and metastasis. So that's one major characteristic of these various clusters. And as you can see that some of these are patient-specific. There are some that belong to multiple, or that come from multiple patients. So this is the cluster identity, and these are the various pathways. Some of these are patient-specific, and some of these come from multiple patients. So there are there are some commonalities as also individual specificities of the patients. The clusters also express genes that are associated with PEMT, partial EMT, which is really a hybrid in the transient stage, and those are the kinds of clusters that we find. These intermediate cell states from epithelial to mesenchymal transition. There is an interesting biology to this. Again, for lack of time, I'm not going to get into that. Predominantly about 41% of the cells are either EMT or undergoing partial EMT, and that's a hallmark of oral cancer, gingivo-buccal oral cancer. We also found that fetal epithelial reprogramming is very, very strong in oral cancer. Fetal epithelial cells are abundant, and as you can see that these are across all of the clusters, there are fetal signatures of these cells, and some of the clusters express very high levels of fetal epithelial cells. The relationships between different malignant cells, again not going to explain to you all of the trajectories, etc., but essentially the malignant cells, as I said, are enriched either in EMT, partial EMT, or fetal signatures, or cancer testis genes. There's one patient who was enriched for cancer testis genes. We did this pseudotime trajectory analysis. There were 4 branches, as you can see. The 4 branches, the B1, which is the basal branch, is populated by cells with enriched signatures of PMT. And B4, which is kind of the late in the trajectory, these cells are enriched with fetal type cells. So we think that PMT state is favoring the tumor initiation, and the cells are eventually transiting to a fetal-like reprogramming or fetal-like state. So the take-home message is that gingivo-buccal oral cancer is governed by 2 dominant cellular programs, one CMT-PEMT, and the other is fetal cellular reprogramming. Of the 12 patients, in 5 patients there is PEMT is the dominant signature, and 6 other patients fetal cell-like reprogramming is the dominant signature. There's one patient in which there is, there was, it was enriched in cells that express cancer testis genes, and this expression is indicative of germ-like epithelial cells. If we look at pathways, malignant cells are abundantly expressed in genes of immune-related pathways. As you can see, multiple clusters are the enriched pathways, antigen processing and presentation, cytosolic DNA sensing, and so on. So it's essentially immune-related pathways that are enriched and abundantly express themselves. The T cells are abundant in tumors with diverse subtypes, and these subtypes are CD positive subtypes, double negatives, and so on and so forth. So there is a diversity of these T cells, but they are essentially very abundant in these tumors. T cells comprise about 37% of these cells, and they occur in multiple clusters, even though there are subtypes of these T cells. B cells are not so prominent. B cells comprise about 7.5%, and plasma and memory cells are the dominant subtypes among the B cells. How well do single cell and bulk cell transcriptomics results agree? They agree quite well, as a matter of fact, and we looked at pathways that are disrupted and so on and so forth. I'll skip this and come back to the similarities between single cell and bulk cell transcriptomics, but essentially the pathways that are enriched as inferred from bulk or single cell are by and large the same. This is more informative. The results from our bulk cell analysis is that these are the genes that are recurrently mutated. These are the genes that are recurrently amplified, and we had homozygous deletion for one particular gene. If you look at single cell results and we look at those cells that are ARID2 expressing, ARID2 is a hallmark of or a characteristic of gingivo-buccal oral cancer, and similarly we look at genes that are cells that express these genes, and if you look at different subsets of genes expressing these different subsets of cells expressing these different genes, what we find are quite interesting. 2 genes stand out. One is the VMP1. VMP1 actually triggers autophagy. The second is NEAT1, which produces a long non-coding RNA that acts as a transcriptional regulator of many cancer genes, and NEAT1 has been found to be implicated in many different kinds of cancers. So we found this interesting, that single cell analysis, as a matter of fact, gave us a little bit more insight compared to bulk cell analysis, in that we were able to identify, among other genes, VMP1 and NEAT1. There was also significant upregulation of CDK6 and DROSHA in these NF1B expressing cells. So we just published this very recently. In September this was published, and that's our paper, first paper on cancers on single cells in a cancer. The lessons that we learned from single cell analysis is that the nature of extensive what we learned is that there is an extensive intra and intertumor heterogeneity in cell types and their proportions. Some of these cell types were, as I said, patient-specific, but many of the cell types or many cell clusters, cell type clusters were across multiple patients. We identified new genes, VMP, well, these two genes, NEAT1 and MALAT1 in malignant cells that influence tumor growth and survival. There's a dynamic nature of the tumor microenvironment that regulates immune response and evasion. Didn't spend too much time on this, but if you look at the T cell, B cell results, this is what comes out. And the expression profiles are distinct between oral cancers with or without association of submucous fibrosis, as I showed you that there were two, essentially these clusters formed two groups, one coming from patients that had this oral submucous fibrosis and the other that did not, which actually did not come out so well in our bulk cell analysis. So single cell analysis indeed gave us much greater insights as to how, the nature of oral cancer, the nature of heterogeneity, and the control of various cell types in oral cancer. Thank you very much.
Thank you very much. We have time for question. Okay.
Hello. Great talk. Great talk. I was wondering, actually, I probably missed that. So, did you look at how the microenvironment of, say, a leukoplakia how does it correlate with, say, the tumor microenvironment or even the tumor adjacent normal? So, do you see any evidence of even field cancerization and the level of?
Honestly, we did not, but in order to avoid field cancerization, we took the leukoplakia sample quite far away from the tumor sample. So essentially to avoid the impact of field cancerization.
All right, great, thank you.
Okay, thank you very much, Partha, one
Thank
more
you.
time. I'd like to now introduce Murim Choi, who runs the functional genomics lab at Seoul National University, and we'll talk about EQTLs and non-alcoholic fatty liver disease. Thank you, Murim.
Thank you. It's a really great honor to present our recent work about NAFLD. I'd like to start my talk with this picture, which shows you how people die by age. So this really nicely illustrates that at the very pediatric stage, there's a big bottleneck really keeps the mutation carriers from living much longer. So this can be a nice presentation of a rare disease. And after that bottleneck, most of the people suffer from chronic disease and die from there eventually. So from there, in our lab, we study rare diseases and also common disease. And the liver, NAFLD, is one of our main projects that I can introduce today. So for chronic disease, as you know, GWAS is the main tool to identify the new genes that are associated with the disease. And I'd like to show that EQTL is also an alternative approach that gives you a lot of different advantages. But I don't have to talk about this in detail for the time constraint. And I just want to make sure that there's a big disadvantage of the EQTL, which is that it shows your minimal clinical implication by itself. So recently, trying to argument that limitation, people has come up with different strategies of EQTL. For example, response EQTL shown here is to separate between cases and controls and separately call EQTLs, and then come up with the EQTL calls that are specific to the cases or controls. Then from there, you can start working on, okay, this signal is patient only, patient specific, and what does it mean? And also another advantage of that approach is that you can separate the patient by genotype so that once you come up with some kind of a treatment strategy, you can identify the patient that with a specific genotype that best respond to that treatment. And the second part, you can have a dynamic EQTL, which shows you that there is a differential EQTL by differing cell status. To going on, today I'd like to further introduce the interactive EQTL, which was already shown in different talks in this conference. And to do that, let me start first introducing DEG, which all of you should know is that by just comparing cases and controls, you can just see what genes are increased or decreased in the patient's tissues compared to the controls. And EQTL, of course, can stratify the changes by genotype so that you can have a much more resolution in that single group. You have a different gene expression differences by genotype. And from there, you have a response EQTL that you have a different EQTL by phenotype. So for example, this shows you that there is a difference in the expression of the certain gene only in certain carriers. In addition to there, what interactive EQTL does is that you have to acknowledge that even patient tissue are composed of healthy and unhealthy cells, and vice versa for the controlled healthy tissues. So that if you further separate controlled tissues or disease tissues by some kind of cell status, low, mid, high, you can compare different kind of gene expression pattern between the two groups. For example, in this example, gene X shows you much lower activation in the TT carriers in the lower. Some kind of cell state, if the cell status is lower in the patient cell, you have a much lower expression in certain genotype. So trying to illustrate how different strategy can give you different resolution in terms of gene expression changes. So NAFLD, I don't have to talk much about it again because previous talks touched on this already. But to try to figure out the novel gene that are associated with NAFLD, we have previously done the bulk response EQTL analysis. So just to giving you the examples, result of that. Taekyung Yoo is my former graduate student who ran this EQTL project, and we got 83 NAFLD bulk RNA-seq data and control bulk RNA-seq data from control individuals, and then ran the response EQTL pipeline to identify the genes that are specifically activated in NAFLD. And then we identified a gene called HCA2, one of the candidates. And then we, after some experimental validation, we were able to see that the HCA2 is an increased, is important for the fibrosis in the NAFLD progression. And then you can basically identify the patient that with carrying the specific genotype that can be best responsive to the treatment against the HCA2, for example. In the next stage, we are trying to expand this study to the single cell level. So Sung-eun is my current graduate student who is running that experiment and who wished to come here, but unfortunately she couldn't make it because she just got married last Saturday and she is somewhere in the Indian Ocean and having a honeymoon, so I couldn't bring her here. So anyway, she has been very, a very smart student to run this order analysis. So we have collected 50-ish samples, half of them non-NAFLD, and half of them are NAFL and early nation, advanced NASH. So this number of 50 is based on our previous simulation that probably we can get at least sensitivity over the EQTL course of at least one, effect size of 1. And then from there, we got about quarter of a million cells. And then this is the UMAP projection showing the nice representation of hepatocyte and the immune cells and other cell types showing here. And as consistent with the previous studies, we show that there is a reduction of one hepatocyte as the disease progression goes on. And then there's a decrease of hepatocyte and increase of the immune cells. And then we ran EQTL callings for the liver EQTL and also interactive EQTLs. For interactive EQTL, we ran Poisson mixed effect model that as following the previous study. To get the cell status, we ran single cell WDCNA and trying to get the gene modules that are specifically associated with a certain meanings of the NAFLD progression. We identified about 12 meaningful modules, and each with some kind of implication with and some of them shown here is that M3 is associated with the fetal liver or adult liver, NAFLD control, or other pericentral or periporal signatures. So our first question is, what are the characteristics of a single-cell liver EQTLs? This is just an overview of our EQTL course, showing the highest number of EQTL called from hepatocyte only, and then cholangiocyte, endothelial, and Sertoli cells. And if you look at the numbers of number of EQTLs and number of number of E genes, and then corresponding number of EQTLs, there's a roughly 1-to-4 ratio across all the tissues that we looked at. And below you see examples of an EQTL showing that there's a nice trend of an expression by genotype. And again, this is the liver EQTL without stratified by cell status or disease. So this is all from the or combination of all 50 tissues. We also saw that our EQTL cores are quite well associated, well correlated with bulk EQTLs or GTEx liver EQTLs. And to look at colocalization, we downloaded UK Biobank GWAS signals from many different traits that are associated with an NAFLD, and we found that among different cell types, hepatocyte EQTLs were most highly colocalized with other NAFLD-related trait. And below shows you just one example of that, shows on a hepatocyte EQTL colocalized with a TRAC2 GWAS signal. Second question was, what are the characteristics of a status-specific EQTL cores, or IE EQTLs? Again, we put the Poisson mixed effect regulation model to call interactive EQTL that can differ by status and phenotype. And this is just one of the examples of such signal, showing that there is a gene called HSD17B13 is nicely separated by HEP8 module, which is implicated with an pericentral gene expression. And what you can see is that there's quite a similar pattern between low and mid, but there's a reduction of expression in the TD carriers in high, meaning that if you think about the liver model and the gene expression in there, there's an increase of expression of the gene from CC, CT, TT, but in the TT, there is a reduction of the pericentral expression. So in actual signal may look like this. Again, this HSD17B13 is already identified from GWAS, and we also saw that this is well colocalized with by EQTL. So in combination, we wanted to see whether our EQTL and interactive EQTL and GWAS, how they were enriched by annotation. And what we saw is that at the very left side, the EQTL is quite highly enriched in active transcription start site, whereas the GWAS signals were highly enriched in the genetic enhancers. And then if you look at the part in between, which shows the flanking regions, the regions flanking active TSS, is there some quite comparable enrichment of GWAS signal and EQTL signals. And interactive EQTL, which is shown as an open circle, are much less enriched compared to the EQTL, which is not really, which was expected by us, because we think that interactive EQTL is a little bit, has a less biological impact compared to the EQTL itself. So also we wanted to compare whether what kind of EQTL best explains the previous GWAS that are associated with an NAFLD. And just looking at ALT level, we saw that all these cell type EQTL best explains the ALT GWAS signal, and then comes the EQTL only from hepatocyte. And this is even higher than the interactive EQTL from across all tissues. Again, demonstrating that EQTL has a much higher impact compared to interactive EQTL. So my thinking is that if you go from EQTL to interactive EQTL, although the biological implication, the quantity of the implication may reduce, it will give you much better resolution of an individualized and cell status level effect. So our final question is that, are there any interesting loci that can be found by interactive EQTLs? And at the same time, we also wanted to answer this question, which is that there's an number of EQTLs. We are thinking that there might be an upstream factor that coincidentally regulating the multiple EQTLs at once. So to answer that question, we were up to searching for module, transcription factor, eSNP, eGene, and phenotype axes. So basically, if there's a transcription factor that is interacting with the cell status, and also if there's an eGene that is interacting with the cell status, that would be very nice. And also, if that transcription factor is regulating the eGene through an eSNP, that would be also nice. So we're trying to look for any axis that satisfy all these kind of criteria. So we come up with a bunch of filtering strategies and using the different algorithms, and we come up with 600 of such axes. And just trying to give you one example, which comes from FOXO1 transcription factor, which is associated with an M12, which indicate a control cell. And then FOXO1 is predicted to influence EFH1 expression through an eSNP that are in the intron of that gene. So first, we saw that FOXO1 is a transcription factor highly expressed in healthy hepatocyte, showing that so this FOXO1 is highly expressed in controls, and M12, as I told you, is highly expressed in the control cells. And then there's quite a nice correlation between the transcription factor activity by CINIC and the M12 model expression. And then there is a SNP in the EFH1 intron, and what we saw is that in the reference earlier, there's it's not supposed to bind FOXO1, but the alternative allele, which changing from A to T, will form a binding site for the FOXO1. And finally, the TT carriers will have lower EFH1 expression, specifically in the healthy cells. So this plot, which I already showed you as an example, showing that in the NAFLD cells, if M12, which is a control cell marker, module, if that's lower, meaning that it's a very unhealthy cells in NAFLD cells, has a very low expression of an TT only status. So that in that, TT carriers, a reduction of an EFH1 is predicted to do something. And indeed, there is a report that EFH1 is a mitochondrial calcium binding protein in the cell survival factor. So it kind of makes sense that reduction of the gene is causing more cell death in the liver, affected the liver. And just in case you guys are curious, I looked at the SNP frequency of the SNP and found out that this alternative allele T is quite low in East Asians, but is actually quite higher in Europeans and South Asians, and Americans and Africans. So I already gave you the example of the first case, and also we found another interesting looking cases of that satisfy all that criteria for the quartet. So another example can be that FOXO1 regulates the expression of a GCGR, which is a glucagon receptor. Meaning, so what we saw is that GCGR was decreased in NAFLD cells in certain genotype carriers. Similarly, PPRG, which is an important regulator that has a direct implication to the metabolic burden, is working as an upstream transcription factor as well as an E gene itself. And we found out that PPAR gamma is regulating more TK and VEGF-A, and also working to enhance the disease by regulating them in the NAFLD cells. And again, EGF-1 transcription factor is working to regulate the PPAR gamma so that the increased PPAR gamma in NAFLD cell can do something bad in the progression of NAFLD. So to summarize my talk, we reported the first instance of a response EQTL on a solid tissue, which yielded an interesting list of genes for NAFLD. And among those, we found that H62 caused fibrosis when decreased, and especially for the certain genotype carriers. And we are working on to the single-cell RNA-seq and EQTL on that. We devised a single-cell level interactive EQTL using human tissues. And then we got a list of functional axes for NAFLD. And I gave you one example, EFHD1 and FOXO1 axis for NAFLD progression. And we are also working on to other interesting looking axes as well. Thank you very much.
Thank you. Any questions? One question?
Hi, nice talk. So I wanted to ask that this study has been done in the Korean population, and we know that PNPLA3 SAM50 haplotype has a strong effect on fat deposition in NAFLD at least, or in other liver disease too. But this particular locus is showing completely opposite effect of the PNPLA3 locus. So have you seen any interaction between these two locus in terms of gene expression at single cell level?
Yeah,
thank you.
That's a very interesting question. We haven't really correlated that with the other signal, but we should definitely look at it. Thank you for the suggestion. Yeah.
Any other questions? One more question.
Murim, I think the single cell interaction EQTL analysis will really open up some new biological insights. Then the interaction QTL anecdotes that you showed were quite spectacular, meaning the effect of cell state was huge. Is that common? Because at least sometimes when you do interaction QTL analysis, across all cell states, the beta is roughly the same. It's significantly different from this cell state to that cell state, but not that different. So how common is it to find such huge interaction EQTLs?
Well, it's really hard to answer that's a very critical question, I know, but first of all, of course, we try to show the best signal that we have. We found that for example, we have a bunch of different modules, and there's always some very best working modules for some reason. We haven't really looked at whether each different module shows a different power, but yeah, definitely that's an interesting way to go.
But do you find that, let's say you compare the effect of the interaction with the beta of the non-interaction SNP, right? Typically, what's the ratio like?
The ratio of in the interacting SNP.
Yeah, versus the effect of the non-interaction effect of the SNP.
I see. I don't think I can think about it right now. I don't think it's really it's dramatically different, but I can tell you right now.
Okay, that's great.
Yeah.
Okay, on behalf of the chairs, we want to thank everybody one more time, all the speakers for this session, and thank you. And thank you, Murim. We'll pass on the chair to the next session.